Am I presenting my screen? Do you see it?
Yeah.
Okay. Okay. ¿Puedo empezar?
¿Tú, Alberto, qué dices? Si no se hace...
¿Empezamos entonces?
Eh, turn off your microphone by a second. Okay. ¿Empezamos, Alberto? De una vez, ¿no?
Sí, ya, bueno, ya, buenas tardes a todos. No sé, ya, eh, ya asumo que hay más gente viéndonos por internet. Muy buenas tardes a todos, bienvenidos. Ya, es un gusto presentarles al doctor Gustavo Sganzerla Martínez, ya que es informático que se doctoró en la Universidad de Caxias do Sul, en Brasil, hace 2 años, ya donde trabajó sobre todo en la notación de elementos reguladores en genomas de arqueas. A día de hoy, Gustavo trabaja de postdoc en el Laboratorio de Enfermedades Emergentes, dirigido por el doctor David Kilvin en la Universidad de Dalhousie, espero haberlo pronunciado bien, en Canadá. ¿Ya? A día de hoy, el trabajo de nuestro invitado se centra en el uso de inteligencia artificial con grandes volúmenes de data genética, ¿ya? Y nos va a hacer una charla que se titula Genetic Information as an Input of Artificial Intelligence Algorithms. ¿Ya? Muchas gracias, Gustavo. ¿Ya? Adelante, por favor.
Bueno, muchas gracias. Voy a cambiar para inglés ahora. No hablo español muy bien. Entonces, good afternoon, everyone. It's a great pleasure to be here. Thanks for the institute for the kind presentation. And in the next moments, we'll be talking about the work that we have been doing here in a partnership with Dr. Ernesto Perez Rueda in the use of artificial intelligence to promote the annotation of large volumes of biological data. Here in this case, we're selecting one particular instance of this data that I'm going to share with you in the next moments. So just before we jump right into the presentation, the biological data itself, I would like to break the ice a little bit and talk a little bit about myself. I come originally from Brazil. Probably the first image that comes to your mind when you when you listen to the word Brazil is not the images that I'm presenting here to you. You probably think of Rio, of hot time in the beach and people are having a good time. Not that we from the South do not have a good time, but the situation is a little bit different with us. For example, the city that I come from is, as you can see here in the map, Vacaria. Actually, we were originally colonized by Spanish people, and as Vacaria was basically the outpost in which people brought cattle from the south of the country up to the northern parts. So the word Vacaria, they used to call it Vaqueria because it was a place where people took their cattle, their cows to rest for a long journey going to the northern parts of the place. We also see here that there are some snowy pictures from my particular city. We do have sometimes cold winters there, so it's not as hot as it is in the other parts of Brazil. This is a common thing that happens in the south of Brazil. And here in the third image, you can see here a lovely picture of a mate or chimarrão, as we like to say. That's a traditional type of tea that is consumed in the southern cone of the American continent, especially in Uruguay, in Argentina, and also the south of Brazil. People say that this is a way to cope with the cold weather, so we do consume these a lot. So that's a little bit about this different part of Brazil that I come from, but right now I am not in Brazil. I am living and working and very well established in Canada, country that I learned how to love a lot. In the particular city here of Halifax, as you can see, this is located in the eastern coast of Canada. It's not exactly an island, geographically speaking. It's an isthmus, as people like to say, But as you can see, due to its geographical location, Halifax has a live-in sea lifestyle in which there's a big fishing industry, and a lot of people have boats. And one good way for people to spend time with their families is just going to the beach, or if you have enough money to buy a boat, then spending some time on the ocean in your boat is actually a good way. This is not as cold as you would expect from the other places in Canada, as you can see here in the map, in Toronto and Montreal and even Ottawa. But we do get some cold winters. However, due to the proximity with the ocean, it's not that cold as it is in the central regions of Canada. These two images that you can see here from Halifax are images that I actually shot these pictures by myself. So I have this hobby whenever I am not in front of the computer, either working or playing games. So recently I got a drone, and everywhere that I go, I like to take the drone with me to try to make some images out of it. Right now I'm in Merida here with Ernesto, and I'm looking for some good places to take my drone in Merida and make some good images to register this beautiful moment. But yeah, that's pretty much it. And now I would like to talk with you about why and what was the path that brought me here today to be giving this talk at UNAM. So I concluded my master's back in Brazil in 2016. At that time, I was like 6 months before my defense, and then my advisor, Dr. Sheila Diavila Silva, once she got me the idea to make my defense as a big event for the University of Caxias do Sul at that time. And we have been using a database from people from UNAM at that time. The database is RegulonDB. One of the authors of RegulonDB is Dr. Julio Cojado Vides. And then at that time, Sheila, my advisor, she told me, why don't we try to invite Dr. Julio Cojado Vides for your defense? Then I said, okay, well, that would be a great way to have the conclusion of my master's there. But I told her, yeah, Sheila, but I do not speak Spanish. And she said, okay, I assume that a guy as big as Dr. Julio is in his field, he might understand English. So why don't you have your defense in English? And at first I was a little bit horrified and anxious about it, but then I got used to the situation, and yes. So in the end of 2018, I got my master's defended with the prestigious presence of Dr. Julio Cojado Vides. He was not present in Brazil at that time. He was virtually. So yeah, we used to have virtual meetings even before COVID. That was already a thing for us. And then we were ready to start that. After my master's was done, I was ready to start PhD. And then we wanted to continue the collaboration because, as we identified here, people from UNAM in Mexico had a great track record, and it would be a great place for us to be at and be collaborators as well. And then we emailed Dr. Cojado Vides at that time and asked if he would like to continue as a collaborator of ours. And then he said that he was taking a sabbatical year at that time. And then he kindly introduced Dr. Ernesto Perez Gueda for us. And then in 2018, we started this collaboration with Dr. Perez Gueda, and we're here today. Almost 5 years have passed, many papers, many publications, and we have a lot going on. So one of the things that I learned with that is, because at first, we were kind of afraid of asking someone as big as Dr. Cojado Vides for being part of our research. And then we said, if we don't actually talk to people, they will never know that we exist. So we basically just went out there and contacted people. One valuable life learning lesson that I took from these is that you are likely to find people that are going to help you instead of people that are going to harm you. And yeah, if there's a way of doing that, just going out there, talking to people, it's a good way to start. All right, but enough with personal and these out of context conversations. Now we're going to have a talk here about science itself. So A little bit of the background in the laboratory that I come from in Canada, in Halifax, is the Laboratory of Emerging Infectious Disease, led by Dr. David Kelvin. This laboratory is associated with the microbiology and immunology department of the institution. So we have a lot going on. I'll talk here about some of the works that we have there. Several clinical studies. We receive clinical samples from different hospitals, partner hospitals around the world, in Europe, in the United States. We process these samples with immunological assays. And me, as being the data scientist of the laboratory, I try to take and produce insights out of these that are clinically relevant. We do have a strong division in bioinformatics as well at the laboratory. There are two postdocs that are only focused in doing bioinformatical analysis. Right now, one hot topic for us, and we're investing a lot of time, effort, and money on, is on single-cell RNA sequencing. So we're using single-cell RNA sequencing as a way to monitor and propose surveillance strategies, especially in countries in Eastern Africa. These countries have been undergoing several endemic species that might be prone to cause a spillover event that in the future might lead to another situation as similar as we got with COVID, for instance. So we have a lot going on in these regards as well. And here I'm showing you an image of a paper that we published some time ago, in which we obtained samples from a hospital. We performed ELISAs to quantify the amount of these immune proteins that are present in blood circulation. And then here, by using machine learning, we could see that we were able to predict the severity of COVID patients by measuring the level of circulatory on these proteins that we have here: ICOM, VCOM, CXCL10. All of these are cytokines that your immune system will express in trying to fight off a pathogen. And we can relate the over and the up or down regulation of these particular proteins as surrogates for explaining severity in immunodysregulated syndromes such as COVID, such as sepsis, and moreover. So this is, in a nutshell, what we have been doing in the lab. We have a lot going on. I could talk about hours and hours on these, but let's move forward. So in another paper that we published on some clinical samples, we analyzed it here that we have three classes of patients with sepsis and sepsis COVID. Both of these patients are undergoing through an immunodysregulated syndrome, but the nature of this syndrome is somehow different. One is viral, one is bacterial. And here on the very end of the table, we also have septic shock patients, which would be the ultimate way of your body trying to fight off that pathogen, that infection that is going to cause you an imbalanced blood pressure, and the mortality rate for these patients is extremely high. But one of the things that we analyzed it here, if we only focus in the sepsis patients, not in the COVID patients, we see here that two-thirds of these patients, they have been indicated as having positive culture. So we have different bacteria in the bloodstream of these patients. And this is an issue that we wanted to address. Here we have some different microorganisms, Staphylococcus, Pseudomonas, and so on. So with that, we try to look more closely at exactly what was going on in patients with these bacteria. And for that, we started to investigate it. So I would like now to bring your attention here to some central processes. So this is what we call the central dogma of molecular biology. Any living organism are going to somehow perform these processes that we have here. We have DNA replication, which is whenever DNA needs to be replicate itself, to give origin to a new cell or whatever. We have the transcription process, in which information stored at DNA level, it gets transcribed into a messenger RNA protein, and then at the end we have that messenger RNA protein being translated through the translation process in a functional protein, all the action of the ribosomes on that. And the process that we want to focus particular here is the middle one, the transcription process, in which DNA is going to give origin to RNA. And for this process to happen, there is the regulation of this process is a tightly regulated series of events that we need to be really aware to fully understand what these organisms are doing. So as you can see here, the very first point for us to start the transcription process is when the promoter is recognized by the RNA polymerase enzyme. RNA polymerase, as the name is already saying, it is synthesized. Its goal is to synthesize a messenger RNA molecule. But in order for it to recognize the particular gene that it needs to transcribe, it needs to be recruited to the correct site on the DNA. And here, as we're focusing specific on bacteria, we need to understand that the organisms that we're talking about have a reduced genome. There's not a lot of space for redundancy. So the more regulated that the transcription process is, that is going to be what divides that particular unicellular organism live in or not. What grants the survivability of an organism also depends on how highly regulated the transcription of the particular proteins that particular cell will need to express in order to have its survivability granted. The way that the recognition of the promoter element happens, then it starts with the recruitment of these blue enzyme that you can see here, RNA polymerase. Then it needs to arrive to be recruited to this particular site of DNA. And if we analyze, at least in a bacterial scenario, we see here that I have two elements here, -10 and -35. And these are the exact points in which these two subunits in this red circle here, this is what in bacteria we refer to as the sigma factor. So this is an assistant protein that will help RNA polymerase to get recruited. The first points of the DNA that this protein will recognize is the -10 and -35 element. And for everyone who works with machine learning, if you have an element that is conserved, that is always going to look not the same, but at least similar, this is the perfect scenario for us to engage on a situation in which we try to obtain a huge volume of data and try to classify that data. So what we want to say here, we'll obtain data from different bacteria, and we want to say what is a promoter and what is not a promoter. So moving forward here, we see here that typically the promoter region of a bacteria, it is going to have these -10, -35 boxes. These are the two extremities in which the sigma factor enzyme, the sigma factor protein is going to attach the DNA. However, that's rather simplistic if you only look to a DNA sequence and tell if that is a promoter or if that is not a promoter, based only in the presence or absence of these boxes here. Here for -10, we have this sequence of nucleotides, the ATAAT, -35, the DGACA. This is for E. coli only, but as it is the well established and studied organism, that's where most of the insights come from. In working with organisms that are totally different from bacteria, Dr. Perez-Gueda and I, we published this paper in 2021, I guess, in which we analyzed promoter sequences in archaea. Those are organisms that likely gave origin to the eukaryotic cells, to multicellular organism. That's why they're called archaea, from archaeological, something ancient. And we analyze it here that it is highly variant, the presence of these consensual elements in the promoter sequences of these organisms. We get here, for instance, Halophorax volcani. This is a specific archaea that thrives in high salinity environments. And we see here that only 1.5% of these promoters are going to have these conserved elements. So if I'm going to look at promoters in this specific organism and try to classify what it is a promoter versus what is not a promoter based on the presence of a TATA box, this consensual set of nucleotides that are present in the here for archaea is around -24, I'm going to fail miserably because, as you can see, most of these sequences of these promoter sequences do not contain the TATA. So by doing that, we wanted to be able to develop a method that would be more reliable in capturing promoter activity rather than only the simple presence or absence of a particular set of nucleotides. And this is when we started to work on our predictor. I will tell you about the data in a bit, but we have obtained it here from some partners that we have from Tezpur University in India. We have been able to get a set of almost 17,000 experimentally verified promoters from 12 distinct bacteria. So this gives us a lot of room for us to try to create a method that will learn what a promoter is or what a promoter isn't. We have here a varied data set of organisms. We have gram-positive, gram-negative bacteria, mycobacterium as well, the different cell wall. So we have a highly diverse data set. And from now on, we're probably going to start talking with more details about machine learning. You know, we are living a particular time in our lives that artificial intelligence, machine learning, this is a very hot topic. I assume that all of you might have heard of ChatGPT and these language models. All of them are based in artificial intelligence. And this is transcending what we see, what we use here, at least in an academia scenario in which we use machine learning, we use artificial intelligence, but only in this highly restricted and controlled environment. Artificial intelligence is basically becoming a part of our daily routines and daily lives. If you access the internet, there will be several different applications of artificial intelligence that are out there, and we live with them. So this is getting more and more attention. And there's a lot of power on artificial intelligence. It can bring us very important insights on varied data. So whenever we're working with artificial intelligence, the key thing that we need to have at first is have reliable data. You know, an artificial intelligence, a machine learning algorithm, it is going to learn if I have good data, and it's not going to learn if I don't have good data. So that's why 80% of the time that we spend doing any type of research and method and any execution that revolves around machine learning, around 80% of the time is only a data pre-processing stage in which we try to insert the data in a well-formatted manner so the algorithm can actually learn, and we can extract meaningful insights on these data. The way that we have done that, as I showed you before, we are trying to transcend what characterizes a promoter sequence, if it has or if it doesn't have the absence of that particular consensual sequences. So we use here one method that we refer to it as a structural parameterization of each promoter element that we have. We're talking here about information stored at the DNA level. The DNA is an alphabet composed by only 4 letters, so it's highly limited. And these 4 letters are always going to be paired in a way that we have adenine and thymine always appearing together, and on the other hand, we have cytosine and guanine always appearing together. So if we analyze the chemical conformation of the combination between these 2 base pairs, ATs and CGs, there will be a difference in the number of hydrogen bonds that make them connected. We have here in pairs between A's and T's, 2 hydrogen bonds, and between C's and G's, we have 3 hydrogen bonds. So by referring to some other studies that we have found in the literature, we can actually measure the amount of free energy that we're going to have here between A's and T's and C's and G's. And by doing that, we're able to represent numerically as part of the amount of free energy that is present in this particular chemical bond. We can represent a promoter sequence not now by letters, but as a numerical representation of them. And in the same paper with Archaea that I mentioned before with you here, we have analyzed the promoter elements of our 3 organisms that we started with. And we see here that if I represent a promoter sequence as a signal of the DNA duplex stability, the amount of free energy that I have between these base pairs, it doesn't matter if that promoter has or doesn't have a data sequence. I can represent the promoter in the same way, whether it has a promoter or not, because this measurement here, stability, it is able to capture this particular relation. Let's focus specifically in this middle image here. As you can see, the lines are basically overlaying. So it doesn't matter if a promoter has the data or doesn't have the data. I can represent a promoter way better by analyzing its physical chemical conformation, in our case, enabled by the analysis of its stability, DNA duplex stability. And we have done that also with bacteria. This is another paper that we published it in 2021. For this one, we focus specifically on bacteria in E. coli. Bacteria, they have a distinct way of getting their transcription happening, which is also the presence of a sigma factor protein that is going to recognize promoters of genes with different functions. And we see here that in each one of the sigma families that we have here, we have a distinct profile. So this only tells us that, yes, we can well represent a promoter element by a numerical representation of it rather than only sequences and A's and D's and C's and G's. And as we're talking here about artificial intelligence, it is great because any application that you're using with artificial intelligence, at some point you will have to convert the data in whatever format that you have, is that numbers, letters, text, speech, images, and you will convert that into some numerical representation of that. And as we are already representing a promoter by a series of numbers instead of a series of letters that we find in the DNA sequencing, so we already have all the means ready for us to get that working in our machine learning. So here I have a profile of the almost 17,000 promoters that I mentioned with you before from 12 distinct bacteria. And we see here that I have a massive peak in the amount of free energy that I have around position -10, -11, that would be somehow around it. And this is specifically where one of the extremities of the sigma factor protein will attach to the DNA. So again, we are able to well represent a promoter sequence if we represent it by its the amount of free energy that you have between its base pairs. Now, moving for the actual machine learning on it, whenever you're talking about machine learning, artificial intelligence, you need to think of a system. In this system, you're going to have inputs, you're going to have the processing, and you're going to have the outputs. The inputs of our system are already taken care of. We have promoter sequences that are represented in DNA duplex stability levels. That's pretty much it. The processing of our system is actually the whole mathematical calculation that we like to get that performed by machines, because it will take a tremendous amount of time if we want to calculate that by hand as humans. And then the output of that system, we can have a prediction, we can have a probability score, a plethora of different things. So what we consider to do here, we tested different algorithms that are able to classify data in a distinct format. Remember, what I am ultimately trying to do here is get a sequence. Is that sequence a promoter or not? That's pretty much it in a nutshell. So we have our promoters. We convert that into numerical values that represent the amount of free energy. And then we use that as part of input for our tool. We have selected here the random forest method. There are several machine learning algorithms that you can choose. We have tested these data in distinct algorithms, and the one that we realized that had the better predictability would be with random forest. Whenever we are trying to do anything in the machine learning environment, we also need to have negative data. The positive data, you already know that are the promoter sequences that we have here. However, the negative data, it's the type of data that I need to insert to the model, so the model can say, no, the sequence is not a promoter. Yes, the sequence is a promoter. So in that particular case, we selected the coding region that comes downstream of each one of our promoters and consider that as the negative examples. So our model can distinguish between the promoter and the non-promoter. In that case, the promoter is the coding region. Other authors, we ourselves have used other methods of acquiring negative data. You can shuffle your sequences. You can get other random genomic regions. In that case, as we hypothesized that only promoter sequences will have a distinct stability profile, then we wanted to have sequences that are not affected by any transcription factor binding to it, such as the RNA polymerase enzyme that I showed you before. So as you can see here, in this particular model, for random forest, we have an accuracy of 83%. So our model is not perfect, for sure, but it does a good job in finding promoters in these sequences. We need to consider here that we have a highly varied data set with 17,000 promoters from 12 distinct bacteria. So we're trying to universally capture what a promoter signal is here. And for machine learning standards, 83% of accuracy, it's not something neglectable. So we decided to move forward with that. But then I stopped and I thought to myself, okay, 83%, but I have 17 every 100 promoters that are not being captured by our model. So I wanted to explore with more details why these sequences were not being classified by the random forest. So we decided to look at the misclassification of our model. And then I need your attention here in this figure B, in which we have three distinct lines. We have the promoters, we have the false negatives, which are promoters that were incorrectly classified as non-promoters. And lastly, we have false positives, which are non-promoters that got incorrectly classified as promoters. So we know that the blue line, the solid blue line, are the promoters. So what we should expect in an optimal scenario is that every promoter will have this peak here around the -10 region. This is what should be expected. If I analyze the false positives, so these are non-promoters that were also classified as promoters, you can see that, yeah, the model has a bias to say that what a promoter is a promoter because of this particular event that is happening here around -10. But then we analyze the false negatives, the sequences that are promoters but were not captured as promoters by our method. And we see here in the orange-ish line that the signal around the -10 position for these sequences, it is still there. There is a distinct signal, but it's not as close as the false positives and the actual promoters that we have. Oh, I'm sorry. So in that case, we also had a statistical comparison in these, and we see that, yeah, these two lines here, the solid blue line and the dotted line, there's no significance between them. So mathematically, I can say that they are kind of the same. However, if I compare the false negatives, the promoters, that we can say that they are weaker, that they do not have that strong signal in position -10, we have a very low p-value indicating that, yes, there is a lot of statistical difference between these. So in that case, we decided to continue with our method and try to increase our model for prediction so we can target only that 17% of promoters that were not correctly classified here by our first model. So we trained a second model. Now this is with an extreme gradient boosting method, which is an XGBoost. This is the one that we identified to perform the best. So we only trained this model with the sequences that were misclassified by the previous model, which was the random forest. So now for a promoter to be classified as a promoter, I will first run that sequence in my random forest. Okay, the random forest told me that's a promoter. That's it, that's a promoter. But if the random forest told me that that sequence is not a promoter, I didn't want to neglect that sequence right away, and I wanted to insert that sequence in a second filter that would be able to capture a lower signal around the -10 position. And that also gave us around 82 to 83% of accuracy. But when we combine these with the other, we try to stay a bit away from the sheer numbers, 82, 83%, whatever, but we need to understand here that we have two models trained with distinct data, and one model is trained with the error of the other. So I am able to capture more varied data in a particular way that is able to deliver better insights to me. So then by doing that, we selected the same promoter sequences that we have started working more than 6 years ago in my master's, published it in RegulonDB, a great database from Dr. Julio Pochado-Vides and other colleagues from UNAM. The current iteration of this database has 3,861 promoter sequences from E. coli. We first got these 3,861 promoter sequences and ran in our random forest model. Here I got, and these sequences were never seen by the model. That's what we call an external validation. Totally new data, and I just wanted to see how my model would react given this new data as input. So out of these, we got approximately 2,000 sequences out of these 3,800 that were correctly classified as the random forest. But as we already expected, I have here almost a 50-50 classification that is not good. If I have, if every 3,861 sequences that I have with these odds ratio here, that's not a good predictor. But then I got the 1,782 sequences, reran that sequence, these sequences in my XGBoost model, and then the XGBoost model was able to capture another 1,464 sequences. So if I combine them both, I have 91.7% of my data that was correctly classified by these two instances. So at first, if I analyzed them individually, 80% of random forest plus 80% of XGBoost, you can easily tell me that, yeah, that there are other works that are able to achieve a higher classification by using a more complex method of classification. But what we try to do here to overcome these is to consider a two-stage classification scenario here. So we have then selected another external base of data in which we had sequences from more than 6,000 bacteria. All of these sequences are only the upstream sequence, meaning that no one has already identified whether that sequence contains a promoter or not. This is something that is still open for scientists to do, and then we propose it to do that. This is published in the RSET tools for prokaryotes, which is also hosted in Mexico here and promoted by Mexican scientists. So we obtained all of these sequences. Now we're talking about 30 million sequences. We selected each one of them individually and then ran in our classification, in our predictor, our random forest and our XGBoost, and we see what we would be able to achieve with these sequences. And in addition, not only the sequence, but also the same tool, our set tools, which is regulatory sequence analysis tools, we also have the functional annotation of those sequences, so we can know what gene might be regulated by that promoter that we're proposing to predict and identify. As you can see here, this was derived by computation analysis using homology. This is a transposase family type of protein. So we are able to combine this information coming from two distinct sources and not only predict a promoter, but also tell what should be the function, what gene that particular promoter is regulating. So with that, we have developed CDB-PROM, which is the comprehensive database of bacterial promoter sequences. We have almost 10 million promoter sequences here from distinct bacteria. So we have here 6,894 distinct bacteria with almost 5 million promoters. And then also in the RSET database, we have different genomes of E. coli more than 5,000 distinct genomes of E. coli, which were also able to extract another staggering amount of 5,300,408 promoters. And what is interesting is that as per the continuous effort of the Universidad Nacional Autónoma de México, here with Dr. Ernesto Perez Rueda, we have been able to publish that data, and everything here is out there. It's in public access, and everyone can have access to these sequences. So here I have in a nutshell how the whole architecture of CDB-PROM is composed of. So remember, machine learning, input, processing, output. Input, we have upstream regions. We want to identify if these upstream regions might constitute a promoter element or not. So we obtained here several, as I told you, more than 30 million upstream regions. This was a high computational effort. We ran all of those sequences in our classifier, and then we have been able to deliver more than 10 million promoter sequences. And this is free for access in this website here. We have some archaeal promoters that we worked in the past, but all the bacterial promoters that we have are here. So you can freely access and download and play around with all of these. We have all bacteria, and here we have a individual folder for E. coli with different genomes. And there's a lot here. That's pretty much it. There's still some work that we plan to do with that. First of all, would be to organize these sequences in a more visually appealing web page, I would say, because let's be honest, the web page that I've just shown you, it is not the most appealing thing visually. I am a terrible front-end developers, so we are constantly looking for people to assist us on that. Also, given all the effort that we did here with CDB-PROM, we also are in continuous effort to develop a web tool in which we would implement our predictor, because right now the prediction itself is only accessible for us. We want any other end user to be able to input their sequences, and then our tool can tell them, okay, based on my prediction, this sequence that you gave me is a promoter or is not a promoter, and so on. And this is a work that is focused only in promoters, but we have all the opportunities as well to be developing for other genomic regions as well, other transcription factors that are not solely on promoter sequences. So that's pretty much it, what I had to say. I would like to thank you everyone for being here with us. Special thanks to Dalhousie University, all the Department of Microbiology and Immunology, where it comes from, all my colleagues, my principal investigator, Dr. David Calvin, also all the people here from UNAM for their continuous effort, and also people from Brazil and Universidade de Caxias do Sul. We have here also the funding agencies that have been in Canada that have been supporting our work constantly. So yeah, that's pretty much it. And again, thank you all for taking your time and listening here to us.
Muchas gracias por su plática, muy interesante el trabajo con inteligencia o machine learning, ¿no? Vamos a pasar a las preguntas que hicieron han pasado. Si tienen alguna pregunta, les doy la palabra a la inteligencia. Está el micrófono disponible para pueda Gustavo responder sus preguntas o cualquier interés que tengan sobre el trabajo que está realizando.
Bueno, sí. como mencionaba, no sé si no me escucharon, los que si se escuchó muy bien, agradeciendo mucho a Gustavo por su plática. Muy interesante el trabajo que ha hecho durante estos años en la Universidad de Dalhousie. Este, Sin más, les cedo las palabras a ustedes para que si tengan alguna pregunta, la pueden escribir en el chat, se puede mencionar para que los lea Gustavo y se les pueda platicar. O pueden optar por levantar la mano y acceder al micrófono para que puedan tener sus dudas o comentarios acerca del trabajo que ha presentado Gustavo. Adelante, Javier.
Hello, Gustavo. Thank you for the talk. My question is regarding your method, if you had a chance to benchmark it against other methods, in particular, if you compare it against just sequence comparisons, like more traditional methods, how does it benchmark against those methods?
Right. Yeah, thank you, Javier, for your question. Yes, so there are, in fact, other methods in the literature that are able to locate these sequences. But one of the things, and I was even discussing with a colleague about it here today, whenever we want to use machine learning, sometimes you have to undergo a lot of layers in turning your data in a more abstract way. So let's say that I am locating a promoter by a simple, let's say, sequence aligning method, a motive identification. The leap that I need to take to convert that type of method into a machine learning approach, it is going to take a series of processes that will require me to modify the input data that I have. And then at the same time, I would lose some of the initial aspects on the data that I have because I had to convert that data into many, many filters. And also one of the advantages of using machine learning is that that's easily scalable. We could here in a short amount of time be able to deliver a high volume of data of sequences that if you would compare that with a traditional method, if you had to annotate anything manually, imagine we're talking here about millions of sequences, which to be achieved by one single person, one single group, one single laboratory would definitely not be.
You have a training data set, right?
Yes, yeah, we do.
So if you take that training data set and just split into parts, can you do a tenfold cross-validation or something like that with different methods and see which one performs better?
Yeah, so actually, yeah, the training data that we have, we divided it in a tenfold cross-validation stage. So our method was executed 10 different times with all of the data that we initially had. So that would allow us to eliminate any bias that would originally be present in the method. So that's pretty much it. We have here some questions in the chat from Paulo Cambranis about the language that I used and the libraries. Thank you, Paulo. I'm going to answer your question in English. My Spanish is really poor, sorry.
I think, ¿cuál es el lenguaje de programación?
I have written this in Python. This is my favorite language. So you asked about the libraries. Then, as I was working in Python, I could use the whole scikit-learn implemented in Python. I'm not trying to say that this is the only way to do that. That's at least the way that I was trained to do so. And I really like Python as a programming language, but I don't see that not happening in any other programming language.
The next
Question is the next question is the free energy data what is best space and uh, the in the free energy data for the best pair what are experimental.
Uh, yes, so yes, sorry about it. Yeah, so the free energy data that we use, this is experimental data that we obtain. So how the original authors developed the quantification of these free energy levels. This is all coming from experimental data. Yeah, we just use their work in conducting ours.
Other question, yes. Did you do other question of YouTube? Did you try other ways of encoding parameters, or one hot encoding, or any other ways to use as test model?
Yes, very good question as well. So we haven't particularly used any other method to encode our promoter sequences. We are aware that yes, you have one hot encoding and other methods as well. They work very well with textual data. However, as we are talking about promoter elements, and it's not that abstract because promoter elements, at least they are expected to contain a higher amount of AT in their sequences. So whenever you look at a sequence that is richer or poorer in terms of AT or GC content, that is going to reflect in the amount of free energy that you have. So this was one of the reasons that we didn't want to include any other method for encoding our sequences that did not represent any biological process. You know, if we're using any traditional method for encoding in machine learning, we would just represent a nucleotide as a series of numbers. Let's say I consider you generally have the moving one, right? So 0001 would be an A, 0100 would be the T. But in that case, this particular configuration doesn't tell me anything about the intrinsic biological process that happened with these. And when, on the other hand, I consider the free energy levels, then yes, this is totally connected with the biological background on that. I haven't used these other methods in our work, but I assume that they would be able to function pretty well, maybe in depicting promoter elements, but we wanted to keep these as close as possible and represent actual biological insights in our data. So that's why we opted to do so. But yeah, thanks for the question. That's a good thing to think about.
The next question of YouTube, we have several question. What about the negative sample? Golden here with the whole presentation. Did you balance your time set, or did you keep actual proportional bacterial genomes?
Yes, thanks for the question. I'm sorry that that was not clear in the presentation. Yeah, so for negative data for each promoter that we have, we selected the direct downstream region, same size it with 100 base pairs for that promoter, so they are balanced in a 1:1 proportion.
Yes, thank you. Other question of Alberto Martin. Did you consider the orientation of the East linear fragment, both forward and reverse, or only forward closer genes, or something like that for both your negative and positive sample?
Yeah, that's a really good question. Thank you, Alberto Martin, for it. Yes, in this particular case, we didn't consider both forward and reverse orientations of DNA. We have only used it reverse, and this is definitely something that we should be able to address in next iterations of our work. That's a good question, yes.
Other question of the people is, what is the effect of the GC content in your prediction? I mean, is if the sequence contains a large proportion of AT or GC, does it have any effects?
Uh, Yeah, very good question as well. This is interesting. So we only hypothesize about these. It's not that I analyzed the capacity of my model to predict only promoters that are GC rich or GC poor. This is something that we unfortunately did not do. But yeah, I see it as a very constructive feedback. But in terms of hypothesizing, we wouldn't expect at least to have a major difference in being able to identify promoters that have more or less GC. But yeah, for me to be able to actually respond to this question, we would need to actually test our model and stratify that into training data with different levels of GC and be able to analyze the prediction for each one of them.
I have other question. How many parameters have you predict for per gene? Only one, or some?
Yes, so we predicted only one promoter per gene. We are aware that we have this is a two-way scenario. We might have promoters that regulate more than one gene, and we also might have genes that are regulated by more than one promoter. This is definitely a limitation of the way that we have our study done right now. However, so far we haven't been able to create a systematic way for analyzing large volumes of data to encompass this type of thing. But yeah, this is something that we definitely should be addressing again in future iterations and thinking about it, because it is already known that we have sometimes promoters that are regulating in case of an operon, or you also have sometimes genes with multiple promoter elements. So that's something that we should be able to find a way to turn that into something systematic. So as we're dealing here with high volumes of data, we would be able to depict this particular feature on our sequences. Great question again.
¿Alguna otra pregunta que tengan ¿Alguna otra pregunta que tengan para Gustavo? Está disponible el micrófono. Unos 10 segundos más para que puedan preguntar. Adelante. Bueno, pues Gustavo, thank you very much for your presentation. Gracias a todos también por asistir a este programa de seminarios. Damos un adelanto, damos por terminado esta plática, nada más para recordarles que dentro de 15 días tenemos también, dentro de la próxima semana tenemos otra plática, tenemos el título de nuestra, una sesión extraordinaria, para que no perdamos el hilo de todos los eventos que hemos tenido. Y le queda de ver la plática de la siguiente semana. No se les olvide, el jueves, ¿verdad? ¿Sí? Ok. 12, 11. El próximo jueves, entonces sería en este canal de Zoom a las 11 de la mañana. Entonces, los que estén disponibles, también se va a enviar las ligas, las invitaciones correspondientes a todos. Nada más que agradecer otra vez de nuevo a Gustavo por su plática, muy excelente trabajo. La parte de los promotores. Sin más, no le queda agradecer de parte. Muchas gracias por su asistencia y nos estamos viendo. Hasta luego.